Well, it's exactly 5 o'clock on the dot on the dial on my MacBook Pro. So, hello and welcome to our Hearing Health Foundation research webinar. I'm coming to you live from sunny New York City, but it's still cold out here. I hope you're wherever you are, it's warm and pleasant. Well, please do enable the Zoom captions by clicking the CC button in the toolbar at the bottom of your screen. Now, these captions can be moved anywhere on your screen. And if you need any other assistance using Zoom, please follow the link to the technical guide shared in the chat. Also, please note that this event's being recorded and will be posted with captions. Now, by way of introduction, my name is Dr. Anil Lawani. I'm a professor and vice chairman for research in the Department of Otolaryngology, Head and Neck Surgery, or ear, nose, and throat, as well as the associate dean for student research at Columbia University, Vagelos College of Physicians and Surgeons in New York. I'm also a board member at Hearing Health Foundation and the head of its Council of Scientific Trustees. Now, the council oversees the Emerging Research Grants program, and affectionately known as ERG, Emerging Research Grants. Now, ERG is a competitive program that awards funds to researchers conducting cutting-edge research in hearing and balance. Now, these grants supported many leaders in our field to become successful scientists, including Dr. Timothy Balmer, our esteemed speaker today. Now, let's get to the topic of our webinar. Our sensory system, including hearing, use two main highways, so to speak, to interpret sounds. There's the ascending pathways that carry sounds from the ear to the brain, and there's also a descending pathway that sends the feedback and contextual signals from the higher levels in the brain back to the lower levels. Now, while many studies have focused on how sound signals travel up through the brain, we're still uncovering how top-down signals improve hearing function at the lower levels. Now, the dorsal, one of the key players in this pathway is the dorsal cochlear nucleus, or DCN, and it's a part of the brain that filters sounds that we make ourselves, like chewing or our own footsteps. Now, it's not exactly clear how the brain does this, that is, block these sounds out. I'm sure we're going to find out from Dr. Balmer today. Now, in this presentation, the Emerging Research Grant scientist, Dr. Balmer, shares how his lab's recent work on this mechanism has shed some light. Now, understanding these pathways in the brain may eventually improve treatments for auditory processing disorders, hearing loss, and tinnitus. Dr. Balmer is an assistant professor in the School of Life Sciences at Arizona State University, where it is nice and warm today. His current ERG grant is in the area of Meniere's disease and is generously funded by the Salus Family Foundation. Dr. Balmer also received ERG funding in 2022 to study Meniere's disease, as well as in 2017 for a tinnitus project that was generously supported by Les Paul Foundation. Now, Hearing Health Foundation's ERG program that provides the seed money, like for Dr. Balmer's work and others, is only possible through the generosity of supporters like you. Now, if you'd like to support the work on hearing loss, tinnitus, and related conditions, you can also do so today at HHF, Hearing Health Foundation, hhf.org/donate. Now, without further ado, we'll move back to Dr. Balmer's presentation. And please do ask your questions through the Q&A linked at the bottom of the screen, not the chat. And we'll try to answer those questions at the end of Dr. Balmer's presentation. Dr. Balmer, take it away.
All right. Well, thank you very much for that kind introduction. I have been supported by the Hearing Health Foundation, as you can see, over the years, and it's been really important to my career success. This 2017 Emerging Research Grant was one of the first grants I got when I was a postdoctoral fellow, and that helped me generate the data that was necessary to get future grants that led to, a faculty position here at Arizona State. So I'm going to tell you a little bit about some ongoing work. This is recent stuff that's not been published yet. It's in review right now, about the descending projection in the auditory system that helps us differentiate important sounds from background. And hopefully I can sort of explain it in a way that everyone will understand and get some takeaway points. Um, in general, the auditory system has a difficult challenge here. We experience environments like this where there's all kinds of conversations going on, and we have to differentiate the signal from the noise. And there are similar frequencies in everyone's voices. So we use a lot of cues based on, experience with conversations, based on the context of the situation, which sounds need to be attended to and which need are just extraneous, irrelevant signals from other conversations. That's a very human example, but if you're a mouse, which is the research subject that I'm using here, you may be walking around on a forest floor that's has these dry leaves, and each footstep makes a crunching sound. And those sounds need to be ignored so that other sounds in the environment can be amplified, like those of an approaching predator. So this is important for an animal's survival. And we think that the brain is constantly producing predictions that help identify the sounds that are most relevant. And it uses other sensory modalities for this. Movement, our motor system moves our skeleton around. And so part of the brain that does the movement can tell the other parts of the brain what sort of sensory response might be produced, like sounds from footsteps. So the brain is interconnected, and we try to think about it on different levels where this higher up level has a more context about the environment that can help improve processing at lower levels from the periphery and the sensory areas. So this is important just for understanding how brains work and how the auditory system works, but it's also related to various disorders, especially auditory processing disorder and various disorders of speech like dyslexia and aphasia. So we've known for a long time that contextual information helps us hear things. For example, this is a study from 1951 that simply shows that when words are in sentences, they're much more intelligible when the same words are presented in isolation. The signal to noise ratio can be quite low, and the word in a sentence can be accurately identified, whereas the same word needs to have a much higher signal to noise ratio. And that's because the sentence tells you what word is likely to come up next. And if there's a couple of words that sound similar, you can get the context from the sentence to guess which one it is. And you'll be right most of the time. You can even skip words and you can guess what was there probably. So that's an example of how in language with humans, we just are able to predict upcoming words and then we hear them better. There's other cues that are not auditory. For example, visual cues help us understand a person's speech better. We use the shape of the person's mouth. We do this lip reading, and that's a visual cue that allows us to differentiate different sounds that sound similar, but are produced by quite different mouth shapes. And this is really important, especially in noisy environments when there's a lot of background, when there's multiple speakers, or when you're listening to a person with an accent. And I think a lot of us realized how much lip reading we do during the COVID-19 pandemic when all of a sudden we were trying to speak to people who are wearing masks. And the masks do reduce the sound a little bit, but I think the main difficulty was that we aren't able to see the person's mouth moving, and that makes it much harder to identify the appropriate phonemes. So we know that there's this higher order information, context that we've learned through our experience of listening to speakers. And that information can guide the processing of the sensory signals. And that requires some connections from these areas that contain this higher order information with the early areas of the auditory system that are processing the bottom-up signals, the signals from the periphery, from the cochlea. So just so we're all on the same page, I want to show this slide. This is from a lecture that I give to my neurobiology undergraduate students, just reminding them what sound even is. Sound, what we perceive as sound, is just changes in air pressure. So if you whack a tuning fork, it vibrates and it produces these concentric spheres of changing density of air molecules, right? And that air pressure change pushes on the eardrum, and that's what produces what we call hearing, sound. So it's this movement of air molecules, and the pressure change, the amplitude of the pressure change is the loudness. That's how much it can move your eardrum. And the frequency of the vibrations of the concentric waves moving in and out is the pitch. So a tuning fork that vibrates really fast will be a high-pitched sound, and a big drum that vibrates slowly will produce a low-pitched sound. So that's what sound is. It's moving air molecules in the environment that's pushing on your eardrum. And I wanted to introduce what noise canceling headphones do. It's not what the auditory system does, but it introduces some concepts that I want everyone to be comfortable with. So noise canceling headphones are actually these really interesting computer devices. They have a microphone that picks up the ambient sound in the environment, and then it produces that same sound, but at an inverted phase. So it's like a mirror image of the sound. And then the speaker moves, and it's actually pushing the air molecules back the way they came. And the air molecules bump into each other, and it cancels out that ambient noise. So this is a form of feedback control where it measures what's occurring in the environment and then produces something that cancels it out really rapidly. And this requires circuits that are extremely fast, little microchip, computer, microphone, speaker. This isn't what the auditory system does. The auditory system can't offset the actual movement of the air molecules, but this does work really well in a computer system like this. So I'm going to tell you what we think the brain is doing, which is perhaps rather more complex than this, but maybe more interesting. So that was a form of feedback control, and usually feedback control is pretty slow. For example, this is from a review that's talking about feed forward and feedback control in the cerebellum, which controls movement. But the concepts are the same in other parts of the brain that are also making predictions about what's out there in the environment. Feedback control is analogous to having a thermostat that turns on a heater when it gets too cold or a cooler when it gets too hot. So what we have here is a desired temperature, which is this range. And when a window opens, it's cold outside in this scenario. And so the temperature drops. And at some point, it triggers a thermostat to turn on the heater. And then the heater comes on, it slowly brings the temperature back up. The thermostat clicks, heater turns back off. But now it's getting too hot. So now the cooler comes on. And so it oscillates across the desired temperature. And the average temperature is just right, but it produces this oscillation. But this is a very simple way to maintain some system like this. What we think the brain is doing is this feed forward control. Instead of waiting until the thermostat clicks and it turns on a heater, it predicts what's going to happen when a window opens and then prevents it from occurring. So here, when a small window is opened, it predicts that the temperature change is going to go like this. It's going to slowly get cooler. And so it applies the heater at the opposite sort of rate. And so it maintains the actual temperature right within this desired range. So this is better. It's faster and it doesn't produce oscillations, but it requires the ability to predict the outcome of things in the environment. In this case, the opening of a small or large window. But we think various parts of the brain are doing this feed forward control. For example, in the cerebellum, which is important for guiding movement and maintaining movements that are smooth. Uh, what the cerebellum is thought to do is predict the sensory consequences of movement. So when I move my arm, my brain is telling the cerebellum what that movement is going to feel like. It knows what the proprioceptors in my muscle will feel like. It tells me what the skin stretch will feel like. And then if it doesn't feel the way that is predicted, then changes occur in order to modulate it so that it does. That's the very brief overview of what the cerebellum might be doing. And I think this feed forward control is also a way to think about what the auditory system is doing. So let's get back to these sort of diagrams of different sensory systems. Sensation begins at the periphery at the sense organs, in this case, the cochlea, the retina, the receptors in the skin, or the taste buds. And you have these ascending pathways. Here's the cochlea. It projects the auditory nerve fibers to the cochlear nuclei, and that goes either through the brainstem or directly to the midbrain, and eventually up to the auditory cortex where perception is thought to occur. All of the systems have these ascending pathways shown on the right and in black, but they also have strong descending projections. So the auditory cortex feeds back to the thalamus and to the inferior colliculus. The inferior colliculus feeds back to different parts of the brainstem and to the cochlear nucleus, and there's even feedback from the brainstem all the way back out to the cochlea. So these are top-down signals. These areas like the auditory cortex and the inferior colliculus have information that the cochlear nucleus doesn't, and it could be providing a guide of what in the environment is important and what isn't. So that's what I'm going to tell you about today. The cochlea processes these acoustic signals and converts them into the auditory nerve's firing rate, which encodes the sound amplitude, and the frequency is encoded by which of the fibers are firing. That's integrated by the cochlear nucleus, which is the main gateway, the only gateway between the periphery and the central nervous system. All of the auditory nerve fibers synapse and are processed in the cochlear nucleus before processing occurs in any other regions of the ascending auditory pathway. So this is a very important place where signals could be attenuated or amplified to improve hearing. Now, the other one I want to point out in this auditory pathway is the inferior colliculus. It receives descending input from various areas and multisensory signals from a bunch of other areas. Signals from the visual system, signals from the somatosensory system. And so the signals coming from the inferior colliculus back to the cochlear nucleus could carry all of this important information that might predict the consequences of movement and other things in the environment that might be background and might just need to be attenuated to amplify the more relevant signals. So this is the pathway I'm focusing on here. So the cochlear nucleus has two parts. I'm going to focus on the dorsal cochlear nucleus. That part is cerebellum-like, meaning that it has a lot of input that is processed by granule cells, which are numerous, and those signals are integrated by a big output neuron. So that's like the cerebellum. And it's thought to act as an adaptive filter. That means that it can attenuate certain sounds, certain frequencies that may be irrelevant, and it adapts to the ongoing environment so it can change what is being filtered. For example, when the mouse is walking around on crunchy leaves, it might want to filter out the high frequency sounds of the crunchy leaves, but if it's slopping around in mud, it might be a different frequency. So it has to adapt to different frequencies to be attenuated. And we're asking where these predictions about what in the environment is background, where do they come from? And so I'm going to tell you about this idea that the inferior colliculus provides those signals. So here's a more anatomical drawing of the ascending and descending pathway just up to the inferior colliculus in the midbrain here. The cochlea sends auditory nerve signals to the cochlear nuclei. The dorsal cochlear nucleus, DCN, projects to the contralateral inferior colliculus. There's a bunch of crosstalk between the two sides of the inferior colliculus. And then there's feedback back down to the cochlear nucleus. And I'm focusing particularly on this feedback projection. And again, the inferior colliculus is a great place that could contain the contextual information that is necessary for guiding the adaptive filter because it receives input from other areas that have more information than even the midbrain has about the context and what might be predictably ignored. We've known about this projection for a long time. Lorente Desnoux, a famous neuroanatomist, drew this in a cat in 1933. These are the descending axons from the inferior colliculus innervating the dorsal cochlear nucleus. And one thing you can tell from this drawing is that the fibers are organized. Some of them are going just to the more ventral part, and some of them are going just to the more dorsal part. And we now know that I think then they also knew that the auditory nerve sends its axons in an organized fashion in such a way that the more dorsal parts of the dorsal cochlear nucleus receive high frequency signals from the cochlea, and the more ventral parts receive lower frequency signals. And so this organization is called tonotopy, and it's overlaid by the descending inferior colliculus projection, which suggests that the descending projection from the inferior colliculus could provide these frequency-specific signals to just the high frequency part of the dorsal cochlear nucleus or just the low frequency part of the dorsal cochlear nucleus, which might be really essential for filtering out specific frequencies. All right, so let's look at the actual data. This is all with mice. And what we've done here is we've injected a virus that expresses a red fluorescent protein in the inferior colliculus neurons. We use these engineered viruses because what viruses do is they put genes into cells. And so we've engineered it so it puts a red fluorescent protein gene into the cells, and then the cells produce it. So it labels them, their cell bodies, and also their axons. And so this is the injection site, but if you look down in the dorsal cochlear nucleus, this is what you see. You see all these fibers coming in and innervating the deep layer of the dorsal cochlear nucleus, not so much in the ventral parts. So we're focusing on the dorsal parts. And this is a very large projection into this area for a feedback projection. So it looks very large, and even the axon terminals are enormous. These are some of the largest terminals that I've ever seen in the cochlear nucleus, in the dorsal cochlear nucleus. And these would be referred to as mossy fibers. They look like moss. And that is a cerebellum-like circuit thing where mossy fibers innervate certain cell types. One of the cell types that innervate are these interesting interneurons called unipolar brush cells. And these are not well understood, but they have a big dendritic brush that is likely to get input from a mossy fiber. They certainly do in the cerebellum. So I'll introduce these and then show you that they do indeed get input from the inferior colliculus. These are important cells because they can amplify signals. They amplify signals in two different ways. In one way, they take in a signal and then they just have these branching axons that end in very large terminals themselves. So probably each one of these unipolar brush cells excites maybe 50 or 100 postsynaptic cells. So it increases the spatial representation of the input signal. But their synapses also amplify. This is a cross section of a synapse using an electron microscope from a paper from 1995, and it shows that this mossy fiber MF, this mossy fiber terminal, synapses on the unipolar brush cell and has this really long synaptic area. So this dark area is the synapses, and it's really long. A normal synapse looks like this one circled here onto a granule cell. But this really long synapse, you only see in a couple of different cell types in the brain, and this is one that has the largest synapses. So what that means is that if you were to record from one of these cells intracellularly and look at the spiking, stimulate the axon, the mossy fiber to release neurotransmitter, it produces a change in firing that lasts for hundreds of milliseconds. And that was the subject of some of the studies I did during my postdoc, and how this synapse, produces these really interesting glutamate AMPA receptor mediated currents. I'm not going to focus on that today, but what I want to show is that these signals amplify signals. So if they get descending input from the inferior colliculus, they would have powerful control of whatever their postsynaptic targets are. So we did this same experiment where we injected the virus to label with red fluorescent protein the inferior colliculus axons, but we did it in a mouse that expresses a green fluorescent protein in a small proportion of these unipolar brush cells. So that's what this looks like. Here's the DCN circled. Those are the descending axons from the IC. Here are the unipolar brush cells. They're all up in there. And if you zoom up on all of their dendrites, you can see these mossy fiber terminals in red, these axon terminals, uh, connected to the dendrite of the unipolar brush cell. So this is anatomical evidence that the descending projection from the inferior colliculus that might carry this contextual information is synapsing on unipolar brush cells. Now we counted all the unipolar brush cells that got input from the IC, and it was at least 10%. And that's quite a huge number, actually, because we only labeled a small part of one side of the inferior colliculi. So probably it could be more like half of these neurons get input from the inferior colliculus. It's actually a pretty big number. So we think that these cells are somehow integrating the information from the inferior colliculus. This was the anatomical evidence, and we wanted to also test the function of these synapses and prove they're, real. So we do this with electrophysiology. This is a really neat technique if you haven't been familiarized with it. It's called acute brain slice electrophysiology. We can take a mouse brain and slice it up into 200 or 300 micron thick sections, and that maintains live intact circuits. And we have really good experimental control. We can apply drugs to block certain receptors. We can visualize the cells, and we can record intracellularly. So this is the electrophysiology rig. Here's a cartoon of what we're doing. We're recording from a unipolar brush cell. We can fill the cell with dye through the pipette. That's what that looks like. And then we can stimulate the axons and do all kinds of interesting things. So that's what we did. We labeled the descending projection again. This time it's green, and we've expressed not just the green fluorescent protein, but a light-gated ion channel. This is called optogenetics. That refers to the genetic expression of this light-gated ion channel, and that allows us to control the activity just of the descending axons from the IC. We injected into the IC, it labeled all those cells, including their axons. So then when we make a brain slice, we can record from a cell and flash the light through the microscope to activate the axons from the IC and see what the postsynaptic effect is. So that's what we did here. We recorded from a unipolar brush cell. If you flash the light 10 times in a row, it produces this big burst of action potentials. So it's amplifying the signal. It's lasting longer than the input here. And it produces these currents. They are direct and mediated by AMPA-type glutamate receptors. So that's our physiological evidence that the inferior colliculus projects to these unipolar brush cells. We also filled them with a dye and imaged them on a different microscope so we can get really high resolution images of the synapse that produce these currents. So we have both anatomical and physiological evidence of these connections. The next question was, what do the unipolar brush cells send this signal to? They're amplifying the signal, but in the cerebellum, which is where we know more about these cells, because they're also present there, they synapse on granule cells. But in the dorsal cochlear nucleus, the granule cells are in granule cell domains, sort of on the outsides, in special areas. There are some in the deep layer, but the unipolar brush cells, there's a huge number in the deep layer, and their axons tend to terminate in the deep layer. So we asked, okay, what are the postsynaptic targets of these unipolar brush cells? So we used optogenetics again. We expressed the light-gated ion channel in the unipolar brush cells. That's what that looks like. It's green and it's in the membrane. And then just to prove that we could make these cells fire with light, we recorded from them intracellularly and flashed light. Here's a 1 millisecond light flash through the microscope. It produces an action potential. We did this maybe 10 times and overlaid the traces, and it produces action potentials with a latency of about 2 milliseconds. And that's going to be important for the next couple slides, produces this big inward current, which is excitatory, and that's what's causing this firing, these action potentials. So we know that we can control the activity of unipolar brush cells with light, and they spike and presumably release neurotransmitter after about 2.5 milliseconds. We then recorded from granule cells, which are the canonical target, the known target of unipolar brush cells. Um, Although they were the known target, nobody has actually shown that they have connections like this in the dorsal cochlear nucleus, especially. So that's what we did. We recorded from granule cells. We flashed the light, which activates unipolar brush cells, and then we measured the intracellular currents. So this is a excitatory current, and the latency with which it occurs is about 3 milliseconds. It's a little longer than it takes for the unipolar brush cell to fire. So that makes sense. That's what a monosynaptic connection looks like. It's about 1 millisecond longer than the action potential itself. We can drive firing in these cells. A single 1 millisecond flash produces action potentials every time. We can drive firing in the postsynaptic granule cell. It fires pretty fast. And so that this is the known connection. So we weren't particularly surprised, but we were excited to show it for the first time this clearly. And so far the circuit that we've built here is that the input from the inferior nucleus innervates unipolar brush cells, and unipolar brush cells synapse on granule cells. Okay, now I'm going to talk about these two other cell types. The cartwheel cell is an inhibitory neuron that is known to get input from the granule cells and inhibit the fusiform cell. The fusiform cell is the main output neuron of the circuit, so the axons that leave the dorsal cochlear nucleus are the axons of the fusiform cell. So no one had ever suggested that unipolar brush cells synapse on cartwheel cells or fusiform cells, but we asked that question because the unipolar brush cells axons tend to end right around these neurons. And so, you know, why not? So we recorded intracellularly from fusiform cells. When you depolarize them, they fire like this. We can characterize them based on their firing rate. And when we flashed the 1 millisecond light to activate unipolar brush cells, we did see the disynaptic inputs that we expected. That's a longer latency, about 10 milliseconds. And that's via this connection from unipolar brush cells to granule cells to fusiform cells, the one that we have known exists. But we also saw these presumably monosynaptic uh, excitatory postsynaptic currents that had the same latency as those granule cells, which are the known targets of unipolar brush cells. So we had this bimodal distribution where a lot of the fusiform cells got direct input from unipolar brush cells based on the latency, and some of them got input that was about twice as slow, which is via the granule cells, so monosynaptic and disynaptic. In some cases, we were able to fill the fusiform cell with dye and image them. And in this case, we can see the green axon terminal of a unipolar brush cell on the proximal dendrite of this fusiform cell, which again gives us the combination of anatomical evidence and electrophysiological evidence of this synapse that people didn't realize existed. So the unipolar brush cells are amplifying these signals directly to the output cell of the circuit. That's pretty exciting. Cartwheel cells, we got the same result. These cells, when you record intracellularly and depolarize them, they produce these interesting complex spikes, so they're very easy to identify electrophysiologically. And we got the same sort of response where some of them were about 10 milliseconds and some of them were much faster, presumably monosynaptic directly from unipolar brush cells. And the UBCs, when we control them with light, they can cause these cartwheel cells to fire their complex spikes when we produce a train of light flashes that gets the unipolar brush cells excited. We were able to identify anatomical evidence of these connections as well. So what I just showed you is that the circuitry of the dorsal cochlear nucleus includes these pathways that we didn't realize, showing all of the latencies of the postsynaptic responses together. The granule cell here is the known postsynaptic target, and that's a monosynaptic input. So the UBC's axon itself excites the granule cell. And we saw in fusiform and cartwheel cells the same latency responses, suggesting that they get direct input. So that's this dotted line to the cartwheel cell and fusiform cell. And they also get a more delayed uh, postsynaptic current, and that would be from the UBC to the granule cell and then to the cartwheel and fusiform cell. So this is all like circuit breaking, figuring out the circuit of the dorsal cochlear nucleus. We're excited about these new results, but we wanted to ask what UBCs are actually doing for hearing and for these cells in an intact animal. So we also have the opportunity to record from fusiform and cartwheel cells in vivo. So we have awake mice walking on a wheel, and we can insert what's called an optrode, which in our case is a 32 channel recording electrode. It has 32 different spots that we hope to get a cell nearby, and an optical fiber where we can introduce the light into the brain, just like we did onto the brain slice with the microscope. Instead, we're just firing a laser through an optical fiber that will excite the unipolar brush cells while we're recording from fusiform and cartwheel cells. Okay, unipolar brush cells are too small to record in vivo like this with this electrode. So in many cases, there was no response to light, but in the following cases, each colored line, we found fusiform and cartwheel cells that responded to the UBC's activation. So here we have everything normalized, but the light occurs for 200 milliseconds, and many of these cells increase their firing rate during the light. So that means the UBCs, which are excitatory, were driving their activity and increasing their firing rate. Okay. We also had three examples of cells that were inhibited by the light. So that suggests that these cells were getting disynaptic input with an inhibitory interneuron in between. So this simply says that in vivo, in a awake mouse that's walking around, we can excite unipolar brush cells, and some of the postsynaptic targets, the fusiform and cartwheel cells, are excited by that, and some of them are inhibited by that. So we know that our laser is working. We know that it's the cells are sensitive to modulation by unipolar brush cells. And then we asked how that affects their hearing. So now we're recording from fusiform cells. I'm showing just the fusiform cells. It looks like this. This is the actual recording. We play different sounds that are random in their order, but different frequencies and loudnesses. So this 6 kilohertz sound at 30 decibels doesn't make the cell fire very much. It's firing along spontaneously. Every downward deflection is an action potential. And 24 kilohertz, 40 decibels makes it fire a lot. 26 kilohertz, 60 decibels also makes it fire a lot. 40 kilohertz doesn't fire much. So this cell has a best frequency of around 24 or 26 kilohertz. And that can be represented in a frequency response map like this, where the y-axis is the sound pressure level, the loudness, and the x-axis is the frequency of the sound we played. And so this fusiform cell's best response is about 25 kilohertz. It responds best there because it responds at the lowest sound level. And it responds to sort of a wider range as you increase the loudness. Now, when we stimulate unipolar brush cells and played all the same sounds, it actually attenuated the response at these lower sound levels. So it was able to filter out some of these frequencies. And when we looked at all of the cells that responded, all the fusiform cells before and after the stimulation of UBCs, we did get a significant attenuation of the response when we're stimulating unipolar brush cells, similar to how we imagine the descending inferior colliculus is stimulating unipolar brush cells. It's attenuating the spiking response to the lower sound levels. So we think that's evidence that this could be guiding the filtering of signals, the acoustic signals in the fusiform cells, which are the cells that are the output of the cochlear nucleus. And this is important because anything that's filtered out at this level in the cochlear nucleus is just not even going to be processed by other brainstem areas, by the inferior colliculus, or be perceived up in the cortex. So this this may be one of the mechanisms for filtering out extraneous sounds and sounds that are predicted to be unimportant. Okay, so here's the circuit that we've figured out. We've added these dotted lines based on our electrophysiological evidence. And we think that these top-down signals from the inferior colliculus could guide the DCN to filter out specific frequencies. And this could be a mechanism to suppress auditory responses that are generated by movement and improve hearing accuracy. Now, for the aficionados out there, you'll say, okay, well, the dorsal cochlear nucleus also gets direct input from a bunch of other areas. Why would the inferior colliculus need to be involved? The dorsal cochlear nucleus gets input from somatosensory areas, from vestibular areas. It gets all kinds of extrinsic input that's non-auditory. But we think most of those signals are going directly to the granule cells. The granule cells' axons go to the molecular layer and they form parallel fibers which innervate cartwheel cells and fusiform cells across the tonotopic axis. So those signals are presumably not maintained in any frequency specific manner. The frequency, if there's a frequency encoded in those signals, it's going to be lost because it is just innervating fusiform cells across the tonotopic axis. So perhaps those signals are non-auditory signals. They could be signals related to the position of the head or the position of the ears if it's an animal with mobile ears like a cat or a mouse. Whereas the signals that need to be maintained as frequency specific, those might need to come from the inferior colliculus because they uh, innervate the dorsal cochlear nucleus in a way that is tonotopic, that matches the tonotopy of the dorsal cochlear nucleus. So that's my explanation of maybe why the dorsal cochlear nucleus gets all these extrinsic inputs, and then also top-down inputs from the inferior colliculus to the deep layers. So I hope what you can take away from this is that there are all these mechanisms of improving hearing in noisy environments and canceling out sounds that are predictable, sounds that should be suppressed, like chewing noises when the jaw moves, footsteps when the feet move. And we're still learning about how the areas that might contain this contextual information uh, improve the processing in these early processing areas of the auditory system. This probably happens in a lot of different areas, but like I said, if it's being canceled out in the dorsal cochlear nucleus, that's really the best place to gate these signals because anything that's canceled here or even just slightly attenuated just won't even be processed in the higher levels. So we have a bunch of other ongoing studies related to this. We're training animals to tell us if they hear tones in noisy environments. And what we'd like to do is make psychometric curves that measure their ability to detect tones in noisy environments. And then we're going to inhibit the descending projection specifically and see if that disrupts this behavior. We're doing more in vivo electrophysiology to try to understand how the inferior colliculus projections are integrated into the dorsal cochlear nucleus and how it affects the firing rates and sensitivity to sounds of the neurons that respond directly to sounds. And we're also working on computational models to predict how this descending projection is related to other disorders, including tinnitus. This feedback loop that I described from the dorsal cochlear nucleus to the inferior colliculus and back is all excitatory neurons. And that suggests that if any of that gets out of whack, you could have hyperexcitability, which is one of the things that we think is producing tinnitus in animals and humans. So we're working on that as well. Finally, I want to thank the people who did the work. This was mostly the work of a PhD student, Christian Candler, but several other folks also were involved, including Kennedy, Olivia, and Isabel. And funding for this includes the Hearing Health Foundation, but also NIH. So with that, I'm happy to take any questions.
Well, Dr. Balmer, that was amazing, wonderful. Really, really interesting to hear about a pathway that we normally don't think about very often since we're so focused on the ear to brain pathway. And
Hmm.
as one might have predicted, one of the two interrelated questions. They're kind of very similar. Is do you believe unilateral tinnitus and hyperacusis are rooted in ascending or descending pathway malfunction? Um, and similarly, Kimberly Churchuk asked, do you have any ideas of how issues with this system might contribute to hyperacusis? And the second part, of course, is there some therapy that you would recommend now that if you think that is playing a role? I guess so. It's a broad three-part question, I guess.
Yeah, so I'm just jotting down some notes here. So unilateral tinnitus and whether it could be related to this pathway,
And
ascending and descending,
hyperacusis,
and
yeah.
hyperacusis. Okay. Yeah, I think about tinnitus a lot because uh there's a lot of ways that this circuit can start firing too much. Too much excitation, not enough inhibition, reverberations, feedback, positive feedback, oscillations. So I I don't know if unilateral is particularly uh, susceptible to this or something, but um, I as I mentioned at the end there, I think that this feedback loop between the dorsal cochlear nucleus and the inferior colliculus could produce this hyperexcitability where the cochlear nucleus is firing a little too much. That makes the IC fire a little too much, which makes the cochlear nucleus fire a little too much. And then it gets out of whack and it starts oscillating and just getting hyperexcitable. Um, and I think this idea, a lot of people have discussed this theory of central gain for hearing loss, where um, when when there is not enough activity, there are homeostatic mechanisms that increase the firing of cells, increase their sensitivity to synaptic input, put more excitatory receptors on them, so they're more sensitive to what input remains. And I think that can cause not only tinnitus because it's more likely to increase the spontaneous firing rate of these fusiform cells, but also hyperacusis because now it's so sensitive. It has become more sensitive, so quiet sounds can be perceived. The loud sounds now are unbearable because it's just, firing too much. That's sort of my, cartoon explanation of that, of the firing. In terms of, there was some question related to whether it's the ascending or descending pathway that is producing tinnitus and hyperacusis. And I think a safe answer is that it's probably both because they're controlling each other constantly. Um, I would love to have some way, and I've been thinking about ways to do this to produce tinnitus in an animal model and then modulate the inferior colliculus to DCN projection and see if it improves their perception of tinnitus. Th That's actually a project I hadn't mentioned that I'm going to be working on over the summer with a group of undergraduates in the lab um, to test the animal's perception of tinnitus and whether it can be improved through this pretty simple mechanism of just turning down this feedback projection. Um, and in terms of therapies, uh, I really have no comment on that because there are a lot of really smart clinicians trying to figure out therapies for tinnitus. Sometimes noise canceling headphones can help people because it reduces that background noise and maybe helps, um, you know, focus the focus on what is most relevant and not background. That might reduce like the processing load, for example. But I don't really have any good ideas of how to improve anything that people are currently doing related to my work, at least.
You talked about how the inferior colliculus, dorsal cochlear nucleus are reducing the sounds you make. You're walking and chewing. Why can't it do the same thing for the tinnitus? It's a question that somebody asked. Of
Mm,
course, I mean, part of it may be that tinnitus arises in more than one station in the central auditory pathway, but can you sort of take it from there and kind of expound on this?
yeah, I mean, wouldn't it be great if it was just doing that? It was just predicting that tinnitus isn't real and then turning it off for us. That would be great. And maybe it is. Maybe what we do perceive as tinnitus is what can't be attenuated by a system like this. So maybe, yeah, maybe if we inhibit this projection in mice with tinnitus, it will actually make the tinnitus even worse. That would be really sort of informative. Um, and it could be explained by that mechanism. Um, yeah, it would be if we could just choose which frequencies to attenuate, that would be great because you could just attenuate the ones that are, if you have like a high frequency tinnitus, just turn it off. Like a lot of the tinnitus people have is in frequency ranges that they can't even hear. So like just turn those cells off. I mean, I'll think about that.
There are some interrelated questions. They have to do with how you can potentially either modify this system or not. But are there pharmaceutical agents that particularly work on the cells you study? And alternatively, are there knockout animal models that give you the lesion in the DCN that allows, or IC that allows you to study what you're studying? So I guess it's both
Hmm.
pharmaceutical and molecular models and interventions that allow you to tease out some of these things.
Yeah, I've thought a lot about this because unipolar brush cells are weird little cells that have receptors that a lot of other neurons don't have. They have these sort of G protein coupled receptors that are somewhat unusual. And, as you know, one of the most difficult things about using a drug to improve anything is that lots of cells have every receptor, uh, or, there's no specific way to get it into a certain part of the brain. So it'll affect wherever it goes in the blood into the brain. And then so so I keep thinking about, okay, these unipolar brush cells, they're excitatory, they're spontaneously active. If we could just turn them down a little bit by increasing inhibitory conductance that is rare in the brain that these cells have, like would that reduce the excitability of the circuits that they're in? Um, that's something I'm excited about trying because they do have like metabotropic glutamate receptors that are inhibitory that are pretty unusual in the brain. So if we could test something like that, it would be really interesting. Record from fusiform cells, measure their spontaneous firing, which is perhaps a correlate of tinnitus, and then modulate their activity pharmacologically. Uh, I think that's something that I'd like to try in the future.
Now, it's not necessarily the focus of your work, but obviously some cells above IC probably control IC's output to the DCN. Your thoughts about which is the dominant? What do you think is going to be the ultimate dominant center? Is it going to be the cortex, or are they all kind of interrelated? Or what are your thoughts about that?
Yeah, there are very large projections from the auditory cortex back to the inferior colliculus. And I think that one is also really important for controlling what the inferior colliculus is doing. Um, I've focused on the cochlear nucleus because it is the area where if anything's filtered out, it just doesn't go any higher. So I think it's the most potent area where this suppression could occur. And also because it's cerebellum-like, it is designed not designed. It's organized similar to the cerebellum, which means that well, to me it means that it's making some sort of predictions that we don't quite understand. The cerebellum is making predictions, cortex is making predictions. Our whole brain is making predictions about what's going to happen in the next like 100 milliseconds because our senses are kind of slow to get all the way up to the cortex where perception occurs. We have to have like a forward model or representation of what we think is ongoing in the environment that we can sort of fit the sensory input to. Um, and so, yeah, your question was about these other areas, and I think the cortex, that's where we have a lot of control as humans to guide like what we're focusing on, what we think needs to be attended to. And so I could see that the cortex is sending signals to the IC that makes it represent the predictions that are even higher than the IC, integrates those with other non-auditory inputs in the IC from the somatosensory system, from motor systems. And then as it's going from the cortex to the IC to the DCN, it gets more and more complex and rich so that it can attenuate whatever signals are, most irrelevant. Like that's what I'm thinking. But certainly the cortex has a huge influence on the inferior colliculus.
So my last question, because you have a lot of questions, by the way, they're amazing questions. I'm sorry to the audience that we won't get to all of them here, but maybe the last question we'll leave with is, where you kind of speculated about the cortex to the IC to DCN. How about the inputs to the cochlea directly? Do these have direct implication for the cochlear or other, feedback systems, either from the MOC or LOC or something that
Hmm.
maybe let's close this complete story, and with that we'll probably end.
Yeah, no, that's a great question. I wonder a lot about what those are doing. The projection out from the brainstem out to the cochlea, which, there's a theory that it's to just prevent hearing damage when there's really loud sounds, but then it's doesn't completely make sense because loud sounds happen really fast and this circuit is not as fast enough to prevent like all of the damage. So it's probably um, it probably has a much more sophisticated role in improving hearing by affecting the cochlear amplifier, the outer hair cells that improve the vibrations of the basilar membrane um, in sort of a frequency specific way. And yeah, I think that's a really interesting question that I know some good people are working on. Uh, but yeah, that's a good another example of this feedback circuit that's improving hearing that we don't quite understand.
Well, I think one of the things that you've clearly done now is we've been so kind of unidimensionally focused on the ascending pathway. You've certainly shined the light on the descending pathway, whether it's all the way from the cortex back to the cochlea, and or in fact, even to the environment with the noise canceling headphones and so on.
Yeah, outside.
I want to take a moment to thank all our attendees to our webinar, and Dr. Balmer, of course, to you for what an amazing, informative session. Thank you for shedding some light on something we don't often think about. We are, of course, grateful to you, our community, for supporting science like the one that Dr. Balmer just talked about. Now, please remember that you can, in fact, donate to our efforts to advance better treatments and cures for hearing imbalance conditions, understanding these conditions, and so on, by donating at HHF or Hearing Health Foundation.org/donate. And thank you, and please do enjoy the rest of your day. And thanks again, Dr. Bummer.
Thanks for the opportunity. I really had a good time. Thanks.